learning outcomes. After studying this module, you shall be able to know about the recombinant DNA technology or gene cloning, learn the methodology underlying recombinant DNA construction, and understand the various applications of this technique. Introduction. In the current scenario, the recombinant DNA technology has found its applications in virtually every sphere of life sciences. Recombinant DNA, also known as rDNA molecules, are DNA molecules that are artificially produced by genetic recombination or molecular cloning of DNA molecules from more than one source. This recombination results in the DNA sequences that would otherwise be absent from their naturally occurring genomes. The basic underlying principle that makes recombination of DNA molecules from different organisms possible is the common chemical integrity of DNA in all existing species. However, even though DNA molecules are chemically similar and follow the basic Watson and Crick architecture, the nucleotide sequences and the overall content of DNA varies between different species. To synthesize a recombinant DNA, a DNA molecule originating from any species can be taken. For example, human DNA may be joined or recombined with bacterial DNA, or a plant DNA may be recombined with a viral DNA. Furthermore, this recombination can also be achieved with artificial or chemically synthesized DNA that may be introduced in recombinant DNA molecules. The applications of recombinant DNA technology are vast and majorly unexplored, but synthetic DNA or any other DNA sequence can be created and introduced into a wide range of living organisms. Discovery of enzymes, the cornerstones of recombinant DNA technology. The key discovery that enabled gene modification was the discovery of restriction enzymes. These enzymes hydrolyze or cut the given DNA molecule at some specific sequences. This was reported by Swiss biologist Werner Arber. They were discovered while investigating the phenomenon of host restriction in bacteria, which acts as a defense mechanism against bacteriophages, that are viruses that invade and destroy bacteria. It was further discovered that restriction enzymes damage invading bacteriophages by cleaving the phage DNA at specific nucleotide sequences known as the restriction sites. This key discovery enabled the biologists to use a tool that can act as a molecular scissor to cut DNA at specific sites as desired or required for subsequent recombination. Another major step was the identification and characterization of another enzyme called DNA ligase. While working with E. coli, an American biologist, Martin Gellert from NIH, that is the National Institute of Health, purified and characterized DNA joining enzyme, an enzyme responsible for the actual joining or recombining the separated pieces of DNA. This enzyme is known as DNA ligase. All living cells use DNA ligase to glue together short strands of DNA during replication. Using E. coli extract, it was further revealed that this enzyme was capable of forming a 3' to 5' phosphodiester bond between the terminal 5' phosphate end of one DNA fragment and the 3' OH end of terminal nucleotide on an adjacent DNA fragment. Methodology: all recombinant DNA protocols are based upon the extraction of genes or gene, that is DNA of interest, from one organism called the donor genome, and then these fragments are allowed to insert themselves individually into bacterial plasmids or any other vector molecule that is capable of autonomous replication within the cell. The vector molecules, in conjugation with the desired insert DNA fragment from donor genome, are called recombinant DNA molecule because they consist of novel combinations of DNA from the donor genome, which can be any organism, with the vector DNA from a completely different source, most commonly a bacterial plasmid or a virus. This recombinant DNA mixture is then inserted into a living system such as bacteria by a process called transformation, where a single or more recombinant vector molecules are taken up by the individual bacterial cells. In order to check whether the recombinant DNA has been successfully inserted inside the bacteria, screening is necessary. For this purpose, a marker gene such as beta galactosidase or an antibiotic resistance gene such as AMP for ampicillin is used. Bacterial cells are plated and allowed to grow into colonies, and those of interest are screened out. If a cell has been successfully transformed with a single recombinant vector, it will divide, giving rise to a colony of millions of bacterial cells which carry the same recombinant vector called as DNA clone. Synthesis of recombinant DNA. The primary step in the construction of a recombinant DNA molecule is to isolate the donor and vector DNA. The DNA extracted from the donor genome has a nuclear genomic DNA in eukaryotes or the bacterial genomic DNA in prokaryotes. Using the commonly available protocols for plasmid isolation, vector DNA can be isolated. However, this may be modified based upon the nature of the vector. Bacterial plasmids are commonly used as vectors, and these plasmids must be purified from the bacterial genomic DNA. This can be achieved by ultracentrifugation by using cesium chloride called density gradient, where it yields a distinct band in the presence of ethidium bromide. The plasmid band thus obtained can be collected by simply puncturing a hole in the plastic centrifuge tube. Another common practice is the use of a specific alkaline pH at which bacterial genomic DNA denatures, but plasmids do not. Neutralization precipitates the genomic DNA, whereas plasmids remain in solution. In addition to bacterial plasmids, phages such as lambda also can be used as vectors for cloning DNA in bacterial systems. Phage DNA can be isolated from a pure suspension of bacteriophages recovered from a phage lysate. Restriction digestion, cleaving the DNA at restriction sites. Restriction enzymes act like molecular scissors and hydrolyze or cut the DNA of the vector at a specific site. It should be noted that restriction enzymes do not cut randomly, rather they cut at specific DNA target sequences, which is one of the key features that make them suitable for DNA manipulation. Any DNA molecule from viral to human contains restriction enzyme target sites at random positions, and therefore it should be able to cut definite fragments of a specific size which is suitable for cloning. Since most organisms lack restriction enzymes, their DNA is not hydrolyzed in natural conditions, and thus restriction sites are irrelevant to the function of the organism. Consider the following example. The restriction enzyme EcoR1 ECORI from E. coli recognizes the following 6 nucleotide pair sequence in the DNA of any organism. The code is 5' GAATTC3' and 3' CTTAAG5'. Such a segment of DNA is called a palindrome sequence, which means that both strands have the same nucleotide sequence but in antiparallel or opposite orientation. A palindrome is a sequence that reads the same in forward and backward direction, such as the English word Malayalam, MALAYALAM. Many restriction endonucleases recognize these palindromic sequences and cut them from a specific point. The enzyme EcoR1 cuts within this sequence and results in a pair of staggered cuts between G and A nucleotides as shown. Such an uneven cleavage is called a staggered cut and leaves a pair of non-identical single-stranded sticky ends. These sticky ends can anneal quickly with a complementary DNA sequence by forming hydrogen bonds. EcoR1 can also be used in making a single cut in a circular plasmid DNA molecule that linearizes it and opens up the circle. The cleaved plasmid also has 2 sticky ends on each end. The principle of recombination through restriction follows that if 2 different DNA molecules are cut with the same restriction enzyme, both will produce fragments with the same complementary sticky ends. When these fragments are present together, they are capable of forming DNA chimeras. Hence, if both the vector DNA and the donor DNA are cut with EcoR1, the sticky ends of the vector can bond to the sticky ends of a donor fragment when both are present in the mixture. A wide variety of restriction endonucleases are known today, which can be used based upon the choice of vector and insert sequences required to produce a recombinant DNA molecule. DNA ligation. When the donor DNA and the vector DNA digested with same restriction enzyme are mixed together, DNA fragments from the two sources can unite based upon probability and availability of complementary ends. Upon joining, these are capable of forming the double helix between their sticky ends. This complementary scenario can result in multiple outputs including vector-vector, vector-insert and insert-insert combinations. The sticky ends, although, have complemented to generate a population of chimeric DNA molecules, the sugar phosphate backbones are still not complete at that two positions at each junction and must be sealed. This important step is achieved by the use of enzyme DNA ligase, which forms phosphodiester bonds at these junctions. This sealing results in a stable vector insert recombinant DNA molecule. Screening and amplification. The ligated recombinant DNA is then introduced into competent cells by transformation. Once a plasmid insert recombinant DNA molecule is in the host cell, it is usually stable and is able to replicate using cellular replication machinery. As a result of replication, the insert DNA, which is a part of the vector's length, is automatically replicated along with the vector. Each recombinant plasmid that enters a cell will form multiple copies of itself in that cell. As a result, in the colonies formed of bacteria on the petri dish, billions of copies of the single donor DNA insert are present. This set of amplified copies of the single donor DNA fragment is the DNA clone. It is also equally important that the colonies be screened for the presence of the righteous, that is, the desired insert, and not just an empty vector. For this purpose, multiple methods can be used. Most common ones are using an antibiotic in the culture plate against which resistance gene is present in the plasmid. If transformation was successful, then only those bacteria will be able to grow into a colony on the culture plate that have the gene encoding for resistance to that particular antibiotic. A second level of screening is made using northern or southern blotting or replica plating technique for confirming the presence of the insert and its sequence. In addition, multiple other methods are available for screening, such as blue-white colony screening that are commonly used by molecular biologists. Applications of recombinant DNA technology. Recombinant DNA is widely used for research in the fields of biotechnology, molecular medicine, genetics, environment conservation, etc. A number of recombinant human protein products have replaced animal proteins and other allied components, for example, the recombinant human insulin and growth hormone. From basic research to the high yielding or insect resistant crop varieties to common pet shops selling glowfish, the recombinant DNA technology has become an indispensable tool with numerous applications. The most significant contribution of recombinant DNA is in the applied biological and biomedical sciences. Recombinant DNA is used to identify, map and sequence genes and to determine their functions. Recombinant human insulin. The recombinant human insulin has almost completely replaced insulin obtained from animal sources, for example, pigs and cattle, for the management of insulin dependent diabetes. The recombinant insulin is manufactured by injecting the human insulin gene into E. coli or yeast, that is Saccharomyces cerevisiae, which then generates insulin for human application. Recombinant human growth hormone HGH, also called somatotropin. Similar to recombinant insulin, recombinant human growth hormone is used in patients whose pituitary glands generate insufficient quantities of growth hormone to support normal growth and development. However, it has also been abused as a performance improving drug by several athletes. Recombinant blood clotting factor 8. In hemophilia patients, the blood clotting factor 8 is missing and thus normal blood coagulation is impaired. This can lead to serious complications including death even by a minor injury. Clotting factor 8 can be administered to the patients to improve their blood coagulation in case of injuries. The recombinant human clotting factor 8 is available for use, which has further lowered down the risk of blood-borne disorders in these patients due to processivity from multiple sources. Recombinant vaccines. A number of recombinant vaccines are now available in the market. One such widely used recombinant vaccine is the hepatitis B vaccine. Common to the third world countries, hepatitis B infection can be controlled through the use of a recombinant hepatitis B vaccine. This vaccine produced in yeast is made up from a recombinant protein that is present on the surface of the virus. This type of vaccine production is a significant advancement for treatment of infections of those viruses that cannot be grown in vitro. Disease diagnosis. Recombinant DNA technology has found its application in diagnostics as well. This is of particular significance in the detection of HIV infection, where some widely used methods have been developed using recombinant DNA. One such method is the ELISA based diagnosis, which uses a recombinant HIV protein to test for the presence of antibodies from the infected person's sample, and the DNA based diagnosis, which screens the HIV genetic material using RT-PCR. Resistant crops. Large number of commercial crops have been genetically modified to give rise to better yielding and commercial varieties of important agricultural crops, including brinjal, cotton, maize, canola, etc. have been developed that incorporate a recombinant gene that imparts resistance to herbicide and simplifies weed control, resulting in better productivity. Several advancements are also being made in insect resistant varieties. These crops are based on Bacillus thuringiensis, a bacterium that certainly generates a protein, Bt toxin, with insecticidal properties. The bacterium has been used in crops as an insect control approach for many years, and this progress has been extensively applied in agricultural science. To summarize what we have learned till now, recombinant DNA technology is one of the most widely used techniques in applicative molecular biology today. Recombinant DNA molecules are created by combining the donor insert DNA with a vector DNA, which is usually a plasmid or a viral vector. Recombination between the two different DNA molecules is possible because of the universal architecture of DNA based on the Watson-Crick model. Recombination of two different molecules could be achieved due to isolation of two enzymes, namely DNA ligase and restriction endonuclease, that join two strands of DNA and hydrolyze DNA at specific sites respectively. recombined chimeric DNA is transferred to a replicating or expression system where its products are realized. Following transformation into a compatible system such as bacteria or yeast, cloned DNA are authenticated for sequence by using screening methods like replica plating and colony screening and or Southern blotting. A wide array of applications for recombinant DNA have been realized in the fields of molecular medicine, agriculture, diagnostics, basic research and many others alike.